G'day, in this video we'll be going over one of the most diverse materials in the world, and that is our soils. In this video we will be discussing some of the ideas presented by Dr. Christine Jones in her presentations called the Secrets of the Sociobiome. Now, with Dr. Christine Jones' retirement, I thought it'd be a great opportunity to condense a lot of those materials and throw in a few other points from myself. As always, there's full credit to Dr. Christine Jones for a lot of the work she's done in the soil space. So if you want to go check out some more of her work, you can go check out amazingcarbon.com and you can see all of her material out there. But otherwise, the aim of this is to really condense a lot of what she talked about. So one of the main ideas from her presentations is that biodiversity is the driver of function within our soil. So diversity, as the spice of life, contributes to nutrient cycling, moisture retention, resistance to pest and disease, especially soil-borne, but also other pest and disease resistance when we talk about the microbes on top of our plants and on our leaves. We'll be talking more about in the soil, but in future videos we can talk about microbes on the plant itself. Productivity and carbon sequestration. Now, it's very difficult to fully understand the complexities of the soil microbes because we can only cultivate less than 1% of the microbes in the labs. And that's because a lot of microbes have these relationships with other microbes in our plants that make them only livable, or they can only survive in the soil. And so it makes it very hard to study these microbes because you can't actually take them out from the soil, put them on a petri dish, and then incubate them and then study them. They all have to be studied in the field. Likewise, we only probably know about 1% of the soil microbes anyways, making it even more difficult to actually understand the full complexities of this system. But this all started with the soil food web as presented by Hunt in 1987. And this is one of the first models or more of the more complete models of how the microbes in our soil interacted and kind of worked together. And this was all based on nitrogen accounting, or calculating where nitrogen is in the system at any particular time. And so effectively the model, this is a simplified version of it, and I'll put a bigger one here, but effectively you have litter or plant litter, residue, animal manure, biomass on the top of the soil surface. What happens is that acts as food for bacteria and fungi. They decompose that, and then different levels of your carbon to nitrogen ratio for each of those, we can talk about later. Anyways, they eat the litter. Then you have these things called protozoa. They're more like hunters. They can hunt down bacteria and some fungi. Also vice versa, some fungi attack protozoa and some bacteria. But anyways, in general, protozoa are the hunters. They graze on bacteria and some fungi. And then because of the nitrogen requirement for protozoa is lower than bacteria and some of the fungi, there's a lot of nitrogen waste, and so that gets released into the soil to be used by plants or bacteria or fungi. You go another step further, we get into nematodes like little worms in the soil, microscopic worms, and they eat protozoa, bacteria, fungi, and potentially other nematodes. And then the next level down beyond that is microanthropods, and then anthropods and earthworms, and then you get the earthworms, you get the birds and the mammals. So it's this whole system of cycling nutrients. And so this whole model is based on decomposition, and that has been revised to consider some of these other pathways that we'll talk about in a second. But the massive missing link is that you have symbiotic microbes that consume root exudates from the plants. And so they're not actually eating, they're not really decomposing material, but more so eating it and exchanging it for other materials. So if you want to call these saprophilic microbes or decomposers, that's good. And the other side, or the others category, is symbiotic microbes. Now, in addition to this, there's actually free-living microbes that don't so much eat litter. They're free-living microbes that produce their own food. There's a few bacteria and archaea organisms that can actually photosynthesize or chenosynthesize, meaning that they can actually take chemicals in the soil and then think of photosynthesizing their own food, but not using light, they're using chemicals in their environment. And the part that is missing is mainly this fungi connection that taps into the plant's that the plants can feed root exudates to, as well as the rhizosphere and the rhizophagy cycle as well. And it's this whole system here where we have a whole range of different plants, a whole range of different microbes, all interacting with each other, is what Dr. Christine Jones calls the soil sociobiome. So as this revised model demonstrates, the carbon captured through photosynthesis and it is fed down into the fungi network, specifically mycorrhizal fungi. Even though there's other fungi groups that form symbiotic relationships with plants, mycorrhizal fungi tends to be the most popular and most common, as well as the most researched. But effectively, this system where the mycorrhizal fungi tap into the plants, get all their food from root exudates, which is, think of a sugary substance that the plant roots produce. If you want to see more about plant root exudates specifically, I have a whole video on that, so you can go check that out here. But this system has been found to be more efficient with its carbon and nitrogen cycling. So some of the main components is that you have your plant, and then your plant root coming down. Now that plant root will produce those photosynthates and root exudates for our microbes. Now you'll have kind of two types. You'll have the mycorrhizal fungi that can tap into, or most of them tap into the plant. So the plant will be feeding the mycorrhizal fungi. Or you'll have the rhizosphere, which is the dreadlocks around plant root. Now both of them will get fed these sugars. We'll dot that to symbolize the rhizo sheath. The plant will feed them sugars to basically use later in exchange for different nutrients or water. But keeping with the mycorrhizal fungi section, what the mycorrhizal fungi also have are symbiotic microbes in relation to the mycorrhizal fungal hyphae. And so similar to the rhizosphere on the root, the mycorrhizal fungi will also have its own microbes to then feed the sugars from the plant. So now you have this whole system where we're capturing light and then feeding it to our microbes directly rather than biomass being slowly decomposed. Now both of these systems are somewhat correct, and you have both of them going on at the same time. But as Dr. Christine Jones talks about, the fungi energy channel is so much more efficient in stimulating microbes as well as delivering carbon to those microbes. And so as she talks about, this is the main channel for building topsoil and sequestering carbon. And if you're interested in building soil organic carbon, we actually have I'm building out a full course for soil organic carbon. So you can check that out either on YouTube here, or if you go to our website, you can check that out. It's completely free and there's not even an email opt-in, which is cool. But one of the main components of that is Dr. Christine Jones's liquid carbon pathway, and you can check that out here. But the two main points of plants feeding our microbes to sequester carbon is that this system is 5 to 30 times as efficient, or more efficient, at building soil organic carbon than the decomposition pathway, which we talked about in the soil food web. And the second point is that this system actually builds soil organic carbon to depth, because you have root exudates being produced as deep as the roots go, rather than say applying compost or even terminating cover crops and it sits on the surface. You really just have the top soil increasing in soil organic carbon. Anyways, back to these interactions. And so you have 3 different interactions going on here. You have plant to microbes, you have microbes to plants, and then you have microbes to microbes. All of which have different interactions, different functions to keep everything in balance. And so this whole thing is about balance and keeping a whole range of different systems in balance so that we can have the most productivity in our soils and our plants. Now these microbes are always releasing what's called auto-inducers, which are little signaling compounds to communicate between the different microbes and the plants, in addition to plant and microbe hormones. That's super important, and we'll talk about that in another video discussing Dr. Christine Jones's work, where she talks about what's called the quorum sensing phenomena in microbes, which uses auto-inducers and certain concentrations of microbes. But effectively, think of this as these microbes and plants are interacting with each other to do particular functions and express different genes. So one of the really cool features is that, say these plants are starting to become deficient in phosphorus, the plants will then signal to the microbes that they need phosphorus. And so then you have microbes that effectively free up phosphorus. And we'll talk about phosphorus as well in another of Dr. Christine Jones's work. But effectively, the plant signals to the microbe that it needs phosphorus. Then the microbe will then mine phosphorus for the plant and then feed it back up through the mycorrhizal fungi network. Pretty cool. And so because of all these different relationships, where you're increasing the nutrient availability to your plant, as well as different hormone signaling and plant growth promotion factors, plants function at their peak when you have a lot of diversity in the soil microbes. And so I'm going to tell you exactly how you can do that in the next part of this video. And ultimately it is increasing the amount of diversity above ground. That will stimulate increased biodiversity below ground. And so what we have here is, I guess, a bit of a comparison between a monoculture crop, I'll just say wheat, and 4 different plants in a multi-species cover crop or a polyculture cash crop. So when you plant a seed, that seed has a whole range of different beneficial microbes that it comes with. That is also why it's very important to get your seed, especially for cover crop, from a really good supplier, where you know the cover crop seed is coming from effectively a regenerative farm or someone who's looking after their soil, because what they're doing will carry on to your farm, the microbes in that seed. Anyways, different plants have different profiles of microbes that they stimulate with different root exudates and different compounds to cater for those microbes. So what happens is when we have lots of different plants, we get these positive feedback loops where you're inoculating your soils with a whole range of different microbes, as well as different profiles in the plant root exudates. Now that diversity of root exudates and communities of microbes is synergistic between everything and ultimately increases the overall complexity of the system, which means when we have more biodiversity, we get more function, and which is what we really want as farmers. We want more function out of the soil because it increases productivity. Biodiversity in our soil microbe community always leads to increase in production. And there's a few studies that actually back this up, and the most prominent is the GINA biodiversity experiment. And what they've done is they've looked at cover crops with monocultures or 1 species, 2 species, 3 species, 4 species, and compared them. And what they found is that biomass is directly proportional to the species diversity of that cover crop. Now there's a few other studies that also back this up. And I looked at a whole range of them during my honours project, looking at multi-species cover crops. And when I was looking at all these different papers, the general consensus or trend I found in these papers was that although biomass is directly proportional to species diversity, doesn't necessarily mean that a multiculture or multi-species cover crop will outcompete a monoculture crop if that monoculture is really high in producing biomass. So for example, sorghum Sudan, like really heavy in its biomass production, summer plant will always outcompete a multi-species cover crop regardless of what you have in it. But what will happen is that if you averaged, say, 16 different plants, so you included that sorghum Sudan, but you also had a whole range of different other things, and then you average the biomass production in that, the multi-species cover crop will outcompete the average of all of those. And that's because you're creating these synergistic effects between them. But the reason why you get this trend of increase in biomass with biodiversity, the first one is that you reduce competition for nutrients at different stages. And it might look like you're increasing competition, but really, if you have a whole bunch of wheat plants going, or monoculture, even a monoculture cover crop, all those plants need the exact same nutrients and water requirements at the exact same stage. Whereas if you have a multi-species cover crop, you have some plants that have a really deep taproot going right down to the subsoil and getting all the nutrients and water from there, where you have some in the topsoil. So those two plants can actually live together with a reduced competition against each other compared to 2 wheat plants that have the same root zone, same water requirements, same nutrient requirements as their neighbour, effectively. And so for that reason, you maximize nutrient utilization and water utilization in the soil. We also maximize light utilization as well. As say you have a C4 sunflower plant growing, and then you have a C3 crop growing as well, or grass. Especially in Australia, where we have really harsh summers, we tend to not use all the light that the sun will provide. And so having the shading effect where the C4s are capturing as much light as they can high in the canopy, but then you have your C3 plants, they're also photosynthesizing. They can take in a fair amount of light, but they require lower amounts of light compared to our C4. And so you can actually maximize your light utilization and therefore your photosynthesis by a range in photosynthetic abilities from our plants, as well as canopy architecture, where you have some plants, say some of the legumes, climbing legumes, can climb up our sunflowers or other plants and increase the amount of leaf space there is. Now, the cool thing about this is that it continuously feeds back upon itself. So increase in photosynthetic ability increases the amount of photosynthase produced, increasing the amount of root exudates produced, which means we can stimulate our microbes more, which then further help our plants, which means you get more photosynthesis and then back and forth. So that was one of the main findings in the GNR biodiversity experiments, was that you get an increase in biodiversity, but there's also a range of other benefits. So you increase microbial biomass, you increase water use efficiencies, you also increase soil carbon and nitrogen capture and sequestration and fixation with an increase in biodiversity. And there's also an increase in beneficial insects and earthworms when you increase species diversity in your cover crop. All of that resulting in just healthier soil, improved structure, all around everything you need to help your next cash crop. Now, there's a few other benefits of biodiversity. The first one is that these plants can actually share the microbes that the individual species bring in. So say you have the sunflower plant bringing in a particular range of microbes, that can share those microbes with, say, your cowpeas. Now, what that means is that under stressful situations, there's a synergistic and collaborative effort between our plants to then survive those conditions. Say during drought, multi-species cover crops tend to do a lot better during droughts or stressful environments than our multiculture cover crops. So with that, when we do have increase in species, we also get an increase in gene pool, which also relates back to our increase in function. So as farmers, what can we actually do to kind of reap all these benefits of biodiversity and the sociobiome? So the first one is increasing biodiversity in our cover crops. So replacing our fallows with a cover crop, or starting to play around with intercropping or companion cropping in our cash crop. Now, Dr. Christine Jones talks about you want a minimum of 4 different plant families in any particular mix. Now, the more the better, but if you want to start, there's almost like a threshold, and 4 is what seems to be where it's at. I mean, 16 would be better, but 4 different plant families is best. Now, it's important that we talk about how you want a range or diversity in plant families rather than species. So if you just have 4 different grass species, so like wheat, oat, barley, and triticale, that's great, and you have more diversity, and that will probably be better than just wheat, but they're all still grasses. They're all going to be somewhat in the same rooting zone. They're all still going to need the same water requirements and whatnot. What we want is increase in actual biodiversity and actual function. And so what we want is one brassica, or this is an example of a cover crop mix. I want a brassica, we want a legume, we want a grass, and we want something from the daisy family or a broadleaf plant, typically the daisy like a sunflower. But broadleaves are a great opportunity to add a lot of biodiversity because they capture a whole range. But say a daisy, what that will do is maximize the overall function of the cover crop and increase that function. Now, all of these plants will tap into the mycorrhizal fungi network. Even our brassica, so non-mycorrhizal plants, can still tap into the mycorrhizal fungi network if there's other plants there to stimulate the mycorrhizal fungi network. You don't want just brassicas because then they don't contribute anything to the network. You want a whole range of different plants. Now, the other thing is that these systems are self-organizing, and with the mycorrhizal fungi network, this plant here can actually trade nutrients, hormones, compounds, whatever, with plants over here. And so there's a few studies looking at different trees, and trees can share nutrients using that mycorrhizal fungi network. And so this system is very much more collaborative than systems where you just have your monocultures. Likewise, if you do have your cash crop, you can put companion crops down, something with, like, say, corn. You can put radishes down and other plants as well. Something to really play around with to reap all these benefits. That's about it with the secrets of the Sociobiome from Dr. Christine Jones. If you like some of this and you want to implement this on your farm, we do provide consulting services at Agresol. So if you want to get started, you can sign up for a free 30-minute consultation with us to see how you can get started. Thanks again for watching. My name is Teal. Cheers.